Okay, so this is general biology, Bio131, and this is part 3 of chapter 6. And this is only a couple of slides remaining. The last time we talked about the GMOs, and we discussed some of the benefits or advantages, pros and cons and disadvantages of the GMOs. This is a list of all of them. You can go through it if you want to. You can make some of these animals or plants that are resistant. You can make some vaccines out of them. You can make it resistant to kills. You can make it resistant to produce herbicides that kill the pits and so on. And you can make more blood supply, food supply or nutrient value. You can make bigger products. The cons are it can have some issues with the health, removing traits, removing the biodiversity. You're getting rid of some of the traits, and so on. So you have this list to go through if you want to, and this is what we did last time. And now, how to fix the genes? Can we fix the genes or not? Like if somebody is having an issue with the genes, can we fix the genes or not? What are the benefits of that, and what's the risk of fixing the genes? Since we discussed lots of details about the genes. So this will bring us to the gene therapy. The gene therapy is if a gene is having an issue, we can try to fix it. It's not successful every time, but at least we can try. And you're doing that to cure a disease or to treat a condition or something. You can try to do that. And what you're going to do is you insert the correct gene in the form of DNA or RNA into that person to try to fix the issue of the defective gene. So this is how it's done. What you're going to do is, let's say this is a person who's having an issue, okay? And we know that this issue is a bad gene, okay? So we diagnose it with a bad gene. So what should we do now? Let's say as an example, this person is having a bad gene and he's not able to make red blood cells, for example, because of one of the genes that are having an issue. What should we do now? Here is what we should do. We pick a person, usually a relative, and we take we isolate a normal gene. What do you mean a normal gene? A good copy of that same gene that's responsible for production of red blood cells, and this is just an example, okay? So let's say just a small error. Do you remember last time when we talked about this replacement, or if you remember that doesn't change much, like point mutation or something like that, something minor. So if that's the case, he's having an issue with the genes and he's not making red blood cells, just as an example. What should we do? We get a normal person and we get the chromosome, the DNA, and we get into that specific gene. So we go deeper and deeper until you get that specific gene, a good copy of it. And then you make a copy of that gene. You isolate it first, and then you use an enzyme that make an RNA version of the DNA gene. So now I have a copy, which is RNA, and this copy contain the good alternative or the good copy of that specific gene. What do you call that enzyme? Does anybody know? What is that enzyme that help you to make a copy of the good gene in the form of RNA? Here is a DNA contain a lot of genes. Okay. I will go to that specific gene only that's responsible for making red blood cells, and I will make a copy of the DNA, and that copy is RNA. What's the enzyme that I told you to remember? RNA? No, it's called mRNA, but what's the name of the enzyme that's helping you to make that copy? RNA polymerase. RNA polymerase is the enzyme that help you to do that. Then what? But are we following the scenario so far? Then what? I found a good person that's having a good version of that gene. I will use RNA polymerase to make a copy of that gene only. Okay, now I have this piece of RNA that have a good copy of the gene. What I'm going to do next, I'm going to use an infectious agent, a virus. This virus can infect, but it's harmless. What do you mean infecting and harmless? Infection doesn't mean disease. Yes.
It means to spread it out, right? But it's not going to hurt the body.
Yes. So infectious, when we hear infectious, by itself, it's not something bad. It doesn't have to be bad. I got infected, so it has to be something bad. No, most of the time it is bad. But infecting means the virus get into your cells. That's it. Okay? We call it infected. Is that going to cause harm or not? This is another issue. Most of the time, yes. But in this case, it is not. So we get retrovirus, and retrovirus is like HIV belong to that same virus, but we don't get HIV, of course. We get a form of virus that's not that does not cause harm. Okay, and we know. Do you remember when we talked about the DNA about the viruses before? The virus itself is RNA or DNA. So we're going to insert this piece of RNA into the virus, and we inject that virus into, like for this example, we inject it into the bone marrow of the person who is having an issue, in the patient. Okay? And infecting that patient. So what's going to happen is this, as we know, the virus, where the virus lives. Like if you're infected with the virus, where does it go to? Which part of your body? Huh? Does it go to the cells? Which cells? Which part of the cells?
The blood cells that need.
Which part of the cells? Not which cells. It can go to any cells. Which part of the cells?
Nucleus?
Nucleus. Yes. The virus go to the nucleus. The virus go to the nucleus and merge into the nucleus. Merge itself inside. Okay? And when it merges inside, the cells are going to start to use it to produce healthy copies. Okay? Did you understand this?
Yes.
How it works? So this is what's going to happen. And it's not that easy, of course, because sometimes it works. Sometimes it doesn't work. So the virus get into the nucleus and it doesn't merge, for example. Or it does merge and a problem happen and this person end up having a cancer or something. So these are the issues. So they are still under trial. Some people actually are cured using this gene therapy treatment. It is promising, but it's not 100% safe. So the issue is some of these kids, for example, that's having issue with the bone marrow, they can be cured. Can happen like this. Sometimes they are not cured, like nothing happened, they can't fix it, and sometimes it can even produce cancer. So these are the issues that they are dealing with, but they use it as a last resort, like the child is going to die. What should we do now, right? He's having severe anemia or something and he's going to die. Or he's not producing white blood cells. Anything can infect him and die, lead to death. What should we do now, right? We should try it. So they try to make it. It's promising, but there are some issues. It can, sometimes it doesn't work, sometimes it have adverse effects. And this is for the gene therapy. Okay?